within a month, we have more than 800 registered participants. These are graduate students, undergraduate students, these are faculty members from universities, these are scientists from all across the place. So basically what we have done is now we have implemented this project as a virtual collaboration, trying to see if we can really achieve something from December to March. And so that is the time frame when most of these graduate and postgraduate students do have time to contribute. So basically the question was, can we create a systems level genetic interaction network for TB? The network that you see here in the center is from East, and we aimed to develop a similar network for MTB. So the day we launched it, we got a lot of criticism, a lot of friction. People say it will not work because it's an untested paradigm. Students don't know how to annotate. You will generate a lot of crap. How are you going to do the QC? There's a lot of managerial overhead. Everything that can come to their mind was thrown back on us. And it was very simple. We just made communities that communicates. So this is a simple solution to all of those problems. If you train well, if you use Web 2.0 applications and tools that helps you talk to the virtual collaborators in real time, if you encourage self-organizing communities, if you can approve community-approved hierarchy and basically motivate them, it can happen, and it did happen. So basically, as you can see here, that a group of students took the responsibility of training freshers. This happened within 1.5 months of online training. So there was a group who was training freshers, there was a group who was taking care of the full text literature access, there was a group who was creating manual for students, and they all did it by themselves. So after 2 months of launching the project, the PIs were thinking if they were anymore required to run it or not, because it was taken over by the community in such a short span of time, and all done by students, mostly students. So basically, instead of following the traditional didactic teaching where there is one teacher and students, we followed a more self-learning in groups, wherein there were team leaders who were teaching the team. So say for example, in my group, there were some 250 members. I had some 8 team leaders. And within 8 team leaders, they were managing some 40-odd students. So that is how I used to interact with team leaders, and they used to interact with their own community members. This is just to show you that how we annotated on a virtual cloud space. You can see that the data is right there. The annotators, you can see they are discussing and what they are discussing. So in real time, they saw the data, they made a discussion, and they took a decision. And given enough eyeballs, all bugs are shallow. So you can see that the errors were marked by the community, they discussed those errors, and they corrected those errors. And this I can skip. The whole point of the take-home message from this slide is that the challenge was not only to get that data that existed almost everywhere, but also to convert it into standard formats like gerontology or SPML. And then we invited the best 100 students from over 1,000 who participated in an on-site phase in April 2010. And you can see the map there which was presented. We could manage to develop that map for MTB in 4 months. And this basically shows you the entire output of the annotation of the 5 themes that I showed you in the previous slide, wherein gene ontology for TB never existed. We have mapped off 4,000, 2,500 RBIDs. We have annotated the structures of MTB proteins. We have identified 262 novel glycan modifying enzymes which work as very good drug targets in other pathogens. We do believe that they will be working as good drug targets in MTB as well. We could identify at least 8,000 immunogenic peptides which were not known before. We created the largest interactome map of MTB with 1,600 nodes and almost 5,600 edges, with 1,200 reactions identified and mined from literature, and more than 2,500 proteins mapped to pathways. So as of today, you have the best annotation on this bacteria available for anybody to download. We are yet to publish the papers, but the data is out from the day we have generated it.
How does this compare with, say, other proprietary databases?
So basically, most of the proprietary databases that you have are either on human, so they give you data on human, or they give you data on other model organisms. MTB is not a model organism to study any of the human diseases. So normally you will not get most of the MTB data anywhere else.
So rephrase that question. How much time will it take a normal entity to develop the same thing? And rephrase Venki's question. Okay. How much time would it take a normal proprietary drug development corporation to do the same work in a linear fashion?
Okay, so I'll just tell you in the next 2 slides how much it will
Okay.
take. So the thing is,
So that's an interesting data for everyone.
exactly right. So I'll just come back to that. So when I compare it to something called string, which is supposed to be the largest protein-protein interaction database that is available, you can see that for MTB there were only 322 nodes as compared to IPW, which has 1600 nodes. And in string, all of the data is computationally predicted, and most of the data that we have has been experimentally verified. And the other details then make no sense because the number of nodes itself was too small. And when we calculated betweenness centrality for each node to basically understand the points where there is maximum information flow in the bacteria, you see that there are some 100 odd stuff. We compared it with human oral, human gut, and the human genome to make sure that these potential proteins do not exist in humans, so that tomorrow if you have a drug which should not go and bind to any human protein, you have at least 50 potential drug targets. And when we compared it to a list that we generated of the drug targets that have been reported till date, the number stands around to 133 as of today. Only 4 are overlapping. So we are at least reporting 46 novel drug targets by doing this analysis, which have never been reported before. So, and this basically also has been ported on PIO, so that we are not saying that the annotation is over and it freezed, because of course that research will go on and you can always value add. So here it is, click on a node, click on an edge, go ahead, annotate it further. You're free to do that, and with your email ID so that you get the due credit. Now this is what it is, Jejit. So the point is that the data that we have collected, we have read papers. So the oldest citation that we have is from 1966 paper. In 2002, there has been a re-annotation study on MTB. After 2002, we have added approximately 2,000 more annotations. Then approximately 1,600 metabolic proteins were assigned functions. More than 600 hypothetical proteins have been assigned functions. Approximately 3,400, which is 85% of the genome, is annotated. And when you say that how much a pharma company will take to do it, so the first thing is, this job has been done for E. coli, it has been done for yeast. This has been done mostly in US based laboratories. There this job is done by postdocs, and many of them over years. And you can now imagine the salary of a postdoc per month and multiply it. And when you compare the cost and the time, so I do not have the slide, but in a recent Nature Publishing Group journal, Bernard Munoz from Eli Lilly, I don't know how he calculated that, he said that Connect to Decode has managed to pack in 300 man-years of work in 4 months. I don't know how he did that, but that is what he has claimed to have said, and it has been accepted. And it has been reiterated in a journal from the Journal of NIH, where they have reiterated the same thing, that we have packaged 300 man-years of work in 4 months. So that is how it correlates to. So pharma companies may never do it.
No, like the $10.7 billion figure that you have.
And when we were doing this entire exercise, we were maintaining a blog, c2d10.blogspot.com, to just make sure that the community is more up to date on what we are doing every day. And this is where we actually had the on-site page in Murthy. You can see that each one of these members who are sitting with a netbook in their lap, this netbook has been kindly gifted to them, gifted to basically OSDD by a lord in UK, Lord Amir Bhatia, and he has basically philanthropized like 114 netbooks from ASA to these students. So that is why people who really recognize the importance of such work give due credits, and they were really happy because none of them actually knew that a laptop is going to be an award for the work they are doing. So they were never told that you're going to get a netbook. So they worked only for motivation, for the sake of work, and not for an award at the end of it. And basically these are other projects that we have been working on to make sure that the community is up to date of where the field stands as of today. So this is basically, you can see the links below and go and find more information. We are identifying all possible literature for what are the reported drug targets, how far they have been worked upon, what are the anti-TB molecules, and now we have launched Connect to Decode phase 2. So it's ongoing, and this time it is not as such a computational activity. Now we are moving ahead to experiments, trying to scale up experiments the same way we have scaled up computational biology, and you can see more on the website. Basically, this is just to make sure that you go and tell to more people that we are maintaining open access repositories for everything that the community needs, whether it is proteins or clones or clinical strains or small molecule. So you just need to go ahead, start your work, don't bother from where you get the materials. We are arranging everything that is required for the community to take forward drug discovery experiments. And either we are acquiring it, we are doing synthesis of small molecules, and there are contributions. Say for example, Loyola College has 2 potential molecules isolated from a plant product which have anti-TB molecules. But for OSDD, you would have never figured it out that there are anti-TB molecules lying in colleges. Just because it is there, they have voluntarily offered that you take it forward to drug discovery pipeline. And this slide is just to show you that with in less than 2 years, we'll be finishing 2 years on 15 September. We have at least one project in OSDD which has moved to lead optimization. This is to give you the status as of 21st August. We have 4,183 registered participants from 127 countries. The second major participant so India is, of course, has the largest blue link, and the red one is from US. So the US people are really following us very closely on what we are doing.
Are they Indians in US or US?
US. So the whole thing is, if together we should fight the problem of TB. And I'm available on all those Skype and GChat IDs. Thank you so much for your attention.